Good afternoon, everyone. While people are starting to come in, maybe I can just introduce this session in general. It's going to focus on brain connectivity, and this is really a critically important topic in the neurobiology of consciousness and cognition. And with respect to consciousness, connectivity often serves as a surrogate for integration, which is thought to be one of the key dimensions of conscious experience, even independently of integrated information theory. This is a theme that runs throughout. And in fact, you've heard earlier today how networks of neurons need to be working together in order to sustain conscious experience. And there are at least two kinds of connectivity, but just to make it easy, starting off, I differentiate between structural connectivity. Those are really the neural highways of the brain, and functional connectivity, the statistical covariation of the activities of different brain regions. And functional connectivity is more like traffic on those structural highways. And I'm sure that we'll be hearing a lot about functional connectivity. We have three speakers today, two of whom are remote, and we are going to start off with Dr. Jean-Rémi King, who is a CNRS researcher at the Ecole Normale Supérieure. He also does research for Facebook AI. And Dr. King's research group is striving to understand the computational bases of human cognition, and to do so is developing a number of new methods and analytic techniques to try to grasp human cognition and consciousness. So without further ado, we can turn it over to Dr. King, who will be joining us remotely, and I thank you all for being here.
Great. Thank you very much for this introduction. Can you hear me well?
Good, yes.
I don't have any feedbacks. Are other people in the first row? Can you raise your hand if you can hear something?
Jean-Rémi, we can hear you and we can see you. Thank you.
Okay, great. Awesome. So I will assume that you also see the slides. I apologize for not being present in person, although I wished this was possible. I would first like to thank the organizers for giving me the opportunity to present our latest work, which are related to consciousness, I think, but perhaps a bit more to intelligence, which is a related notion, but not directly identical. And given the diversity of this crowd, I thought it would be entertaining to argue in favor of a controversial hypothesis, or at least to present you with some empirical evidence that fuel a debate related to a controversial hypothesis, which is this hypothesis that there may exist some general laws of intelligence that explain how neurons integrate information with one another and ultimately generate functions which are useful to understand how the world works and how to adapt behavior accordingly. Now, to many of you, this may not sound like such of a controversial idea. After all, our disciplines from psychology and neuroscience are tasked to identify the regularities of phenomena, to organize these regularities, and to understand the underlying principle, the underlying laws that explain such regularities. So what's so controversial behind this idea is the idea of generality, and to try to capture, to crystallize the controversy as much as possible, I thought it would be useful to turn to a tool which is known to make anything controversial, namely Twitter, because it's always good to point the finger to other companies. So here goes a tweet from Timai, the AI guy, who asked Yann LeCun, who is the recipient of the Turing Prize and one of the founders of the latest version of deep learning. Timai, the AI guy, asks, any takes on general artificial intelligence? And Yann LeCun says, it does not exist, general intelligence, artificial general intelligence. Even human intelligence is highly specialized. We can talk about human level AI, but AGI, artificial general intelligence, is nonsense. So let me unpack this tweet, if I may. There are really two ideas, right? On the one hand, which is something which I think pretty much everybody would agree on, we observed a wide variety of natural species with whole characterized by specialized intelligent functions, and from the spider to humans, obviously there are huge differences. And on the other hand, and this is the second part of the argument, this wide variety of behavior of intelligent system goes against the idea that there would be a unifying law, a unifying set of principles underlying intelligence. And we've heard, I think, this kind of logical arguments throughout the history of science, right? The variety of movements that we observed in objects here on Earth or in the skies is so high that really it goes against intuition that there would be a general law of gravity. The wide variety of species that we observe in nature is so high that really it does not seem possible that there would be a law of evolution by natural selection. And perhaps more recently, the variety of languages that we can hear across the world is such that it is difficult to believe that there would be a universal grammar, a set of syntactic rules that are shared across individuals. And so in this talk, I will not really defend this possibility, but I will present you with the method that we follow in the lab to try to come about this issue, to try to understand how the neuronal systems, especially in the human brain, integrate information in a way which achieve intelligent function, and to try to see whether this approach allows us to identify general principles that would be shared independently of the species that we are studying. Okay, so the approach that we undertake is pretty standard in the field nowadays. It consists here in comparing two vastly different types of agents. On the one hand, we have the human brain, and on the other hand, we have a deep neural network trained to do a particular task. And in this study and in this approach in general, what we do is we try to see whether the representations which are being learned by the model correspond to the representation in the human brain, with this idea that any similarity or any differences would highlight whether or not indeed we can identify some common principles between the two. So the schema that you see on the top here corresponds to a deep convolutional neural network stacked with a so-called transformer. The details don't really matter. It's a pretty standard deep neural network nowadays, which was here developed by Alexei Baevsky at Meta, Alexei who joined our team recently. And it's a deep net basically trained to do self-supervised learning on speech data. So you provided this network with many hours of speech audio input, and it basically learns how to extract from these speech inputs representation which are useful for predicting parts of the data given the context. The question that we ask is, are the representation generated by this kind of algorithms similar to the representations generated in the human brain? This is a set of data that we are looking at, which is created from a bunch of different data sets and which consists of 587 subjects recorded with functional magnetic resonance imaging while they were each listening to about an hour of audio stories. Okay, so to compare the representations of the deep net and the representations of the human brain, we basically want to compare their activations in response to the same inputs. The idea is that we want to see whether the activation of the deep net that we will call X are in any way similar to the activation of the brain that we will call Y in response to the same speech data. Now, because there is no obvious one-to-one mapping between each artificial neuron in the model and each biological neuron in the brain, we need to learn a so-called mapping, which is simply here a linear regression. And you can think of this mapping as really a linear readout. And so you can think of this as a virtual neuron, which is added on top of the algorithm. And we want to see whether these virtual neurons behave in any way similarly to the brain when they are inputs with the same type of data. Okay, so let me show you a brief illustration of what we're looking at. Here on the horizontal axis is the time that unfolds from the beginning of the stories, of an audio story. And on the vertical axis is the amplitude of the bold activity of the brain response as measured with fMRI. And the black line corresponds to the true measurement in the fMRI. And the red line corresponds to this, the red line corresponds to the virtual neuron, to this linear readout of the activation of the deep net. And so what is striking here is that while the two lines are not perfectly matched, they are pretty much similar. If you were to correlate the red line and the black line, you would find a highly significant correlation, which means that this virtual neuron here has the same sort of function as this, the ensemble of neurons which is recorded in this particular voxel, which is being cherry-picked here, in that it responds in a way which is similar to this ensemble of neuron picked up by this voxel in fMRI in response to the same speech data. And so this approach really gives us a simple method to compare the representations of a deep net and the representation of the brain. And all of this is based on basically on a linear correlation between the activations of the deep net and the activation of the brain. And so the brain map that you see over here corresponds to the systematic analysis of the correlation between the deep net and each voxel. So for each voxel, we can compute this correlation between the virtual neuron and each voxel. Okay, and so what you see that it peaks around the auditory regions, but it's pretty much significant across the entire cortex at some rare exceptions. And although it's a bit dim, it basically encompasses the speech networks typically associated with speech tasks here. Okay, so we have a method that can show or can quantify the extent to which the representations of a deep net are similar to the representation of the brain. What is even more striking is that we can evidence with this method that the hierarchy of representation which are being learned by the deep net corresponds to the hierarchy of the language network in the brain. For this, we do a very simple analysis. For each voxel, we try to see whether its correlation with virtual neuron is maximal for the superficial layer, for the middle layer, or for the deep layer. We can estimate which is the layer in the network which best corresponds to the response profile in this particular voxel. And what we observe overall is that the early auditory areas are most similar, I should say, to the most superficial layer in the deep net, whereas the prefrontal areas and the superior temporal gyrus are best correlated with the deeper layers in the deep net. And that's very important, right? Because it suggests that this deep net has learned a hierarchy of representations, has learned to integrate information in such a way that it actually matches the hierarchy of the cortex here for the language task. Now, this correspondence between the hierarchy of the deep net and the hierarchy of the brain is not only visible spatially with an anatomical analysis like you see here, but also temporally by using a different method. This is something that we've shown together with Charlotte Caucheteux, who is a PhD student, co-advised by Alexandre Grandfort at INERIA and myself. Charlotte employed the very same technique. She presented two DeepNet, this time it's a visual DeepNet, VGG19, a series of words which were actually presented to human participants while their brain activity was being recorded with both functional magnetic resonance imaging in a given session and magnetoencephalography in another session. And she compared the extent to which the activations of the DeepNet matched or were correlated with the activations of the brain in response to the same word presented visually. So the result of this correlation analysis is the following. Here is the average correlation across 100 individuals that were collected by Jan Matthias Schöffelen at the Donors Institute. You see that the blue patches here are the regions which are significantly correlated, meaning that their response profile elicited by each individual words correlates with the response profile of the virtual neurons extracted from the DeepNet. And this corresponds basically to the early visual areas. Now we can do the same with magnetoencephalography and find, although a more blurry picture, something which is consistent with this finding. So perhaps for those of you who are not used to magnetoencephalography, it's a neuroimaging device which is not that popular because it's pretty demanding in terms of processing. But this device measures the fluctuations of magnetic fields elicited by the postsynaptic potential of pyramidal cells in the cortex. In practice, when pyramidal cells fire, they create an electric current which is associated with an magnetic field. And because pyramidal cells are aligned with one another in the cortex, these electric fields and magnetic fields can be picked up by distant sensors which are located outside the head. And we can do this measurement pretty much every millisecond, which allows us to have a high temporal resolution of the brain activity. And in this particular task, what subject did is that they read sentences one word after the other. So we have once upon a time, each word is a flash one after the other. And for each word, we can therefore measure the brain response millisecond after millisecond in response to those words. After some pre-processing for each individual subjects, we can identify the extent to which each brain region is more or less activated by a given word. And we have to take this source estimate from magnetoencephalography with a grain of salt because it's not perfect, but we can corroborate these source estimates with intracranial recordings on which we apply the very same analysis. And overall, we find the similar type of response profile. It starts from the early visual areas, it propagates very quickly along the temporal and the parietal areas, and quickly reaches the prefrontal cortex, ultimately end up in the temporal and inferofrontal areas. And this is something that we observe pretty much, although with some variation, with a very similar dynamic in intracranial recordings, here collected in 176 patients implanted for clinical purposes. Okay, so what you see here is actually the average evoked response to individual words that are being flashed one after the other. But we can again analyze these brain responses at the single trial level to see the extent to which at the single trial level these activations correspond to the activations of a DeepNet, and see whether these two sets of activation, the brain and the DeepNet, correlate with one another. So what you see over here is a result of that correlational analysis. The time unfolds over here, and we see that around 100 milliseconds after the onset of the word onto the subject's retina, we have the back of the brain, the early visual areas, which starts to have activations which correlate with the activations of here VGG19, a deep net trained on image recognition. Now, this correlation that I've shown you is actually with one deep net. This is a deep net trained to do object recognition, or more specifically here to do character recognition from natural images. So its goal is really to transform pixel input into letter input. And the representations that it generates seem to correlate with the early parts of the visual system. But we can do the same type of analysis for different types of deep nets, for different types of algorithms. For instance, we have word embedding algorithms, which are tasks to find vectorial representations of words which are maximally predictive of context. Or we can take algorithms like GPT-2 and now the subsequent version of GPT, which are so-called language transformers. And these are deep nets which are trained to predict the words that will come up after a given context. And what we observe overall is that different deep learning algorithms generate representations that specifically map or specifically correlate with different brain areas. We have the early visual cortex, which is most similar to the representations of the VGG19, a perceptual convnet. We have the left temporal regions and prefrontal regions, which are most correlated with the word embedding, word to vec, and we have a very distributed network, which is highly correlated, or at least relatively correlated with a language transformer here at the equivalent of GPT-2. But again, we can do this analysis not only in space, like I show you here, but as a function of time. So I showed you in the previous slides what happens within the first 100 milliseconds. Within the first 100 milliseconds, we have a correlation between brain activity and the and VGG19 in the early visual cortex. This effect does not stop here. Actually, it continues along the dorsal stream here and the so-called ventral stream. Around 200 milliseconds after the onset of the word onto the subject's retina, this correlation here turns into cyan. Cyan is actually an overlap between two effects, between a green effect and a blue effect, between the correlation of brain activity and word to vec, a word embedding, and a correlation between brain activity and VGG19, a perceptual convnet. And this is particularly interesting because this area is has been for a long time predicted to be the area that transforms visual input into the lexicon. It is a so-called visual word form area that has been heavily studied by Laurent Cohen, Stanislas Dehaene, and a few others. So this transformation between visual representations into lexical representations seems to occur here around 200 milliseconds after word onset. And after that time period, we start to observe this rise of green color here in the left hemisphere, which is at the moment absent from the right hemisphere, at least the lateral parts. And this really peaks around 400 milliseconds. And this indicates that all of these areas now have activations which correlates with the representations of word embedding, which again is very consistent with what we know, because we believe from decades of electrophysiology and EEG research that 400 milliseconds corresponds to the approximate amount of time necessary to retrieve the meaning of word, to retrieve a wide variety of features related to each word, which presumably is encoded here in the left hemisphere. After that time period, there seems to be a lot going on. There's a lot of recurrent activity. It's not quite clear what's happening. I must say there's probably a lot to be done there. But after about 800 milliseconds, we start to see rises of red color, and ultimately the whole brain actually turns red. And what that means is that, at least what this indicates, is that now the brain activity in each of these regions, the neural activity that we can pick up, it starts to not being correlated with the deep convnet, VGG19. It starts not to be correlated with word to vec anymore. It's only correlated with language transformers like GPT-2. And so it seemed that now we have a method to really track the rise of different levels of representations which are similar to visual neural network, similar then to word embeddings, and ultimately similar to what we call language transformers, which are network which can combine the meaning of words together, that can integrate their different features in a way which is more than the sum of their parts. Okay. So I've shown you that three pieces of evidence. First, we have a simple method to quantify the similarity between the representations of brains and algorithms, and it seems to be clearly above chance level. Second, I've shown you that the hierarchy of representation that artificial neural networks generate, at least the one that we've studied so far, this hierarchy corresponds to the hierarchy that has been evidenced over the many years and decades of research in language and in perception. And finally, I've shown you that the sequence of representations generated in the brain matches the sequence of representations generated in the sequence of algorithm, going from perception and ultimately to the integration of multiple words with one another. Now, in this last part, I will show you that the key factor that predicts the similarity between a deep learning algorithm and the brain is the loss function. So the loss function of a deep net is really what we tell the algorithm to do, right? So some algorithm, we train them to do object categorization. The loss function here is really accurate categorization. We say, if you say it's a banana and it was a house, you get it wrong. If you say it's a banana and it was a banana, then you get it right, and you are reinforced into making the same type of function, the same type of representations. In other types of algorithm, it's different. We say, try to guess what the next word will be, and if you're correct, we'll reinforce your parameters to force you to give a similar function, and if you're wrong, we will tell you to try to find a different type of function. And so the loss is really the objective that the algorithm is trying to achieve, and what I'll show you is that the loss here seems to be the key factor, the key predictor of whether or not an algorithm is similar to the brain. So how did we do that? In practice, what we did that we retrained many different types of architectures and algorithms. We varied the depth, the size, the number of attention heads, the number of parameters, the number of dimensionality, the amount of training, the amount of data for each of these algorithms. And basically what we ended up with is this map, where on the horizontal axis is the ability of the algorithm to predict what's going to come next, given the past. And on the vertical axis is how similar the representations of this deep net is, given how similar the representations of the deep nets are to the brain. And each dot here corresponds to one artificial embedding, average across all voxels in the brain or all MEG sensors and all subjects, just for clarity purposes. Okay. And so what we see overall is a pretty striking correlation. The better the algorithm is at predicting words from context, the more its representations tend to be similar with brain activity. And that's, to me, a very important finding. It suggests that, at least in deep learning, in artificial intelligence, we are on the right path. If you make an algorithm that can do something right, that can predict the right future, then this algorithm will naturally generate representations which are similar to those of the brain. Now, if we do some statistical analysis of all of these different experiments, what we observe is that this ability to predict what's going to come next, given the past, is actually the highest predictor of whether or not an algorithm will be brain-like or not. Whether the algorithm has a high amount of dimensions, many layers or not, the type of task that we've been using it for, even the amount of training that we provide the algorithm with are relatively modest predictors of whether or not an algorithm will be more or less brain-like. The key factor that makes it brain-like is how good it is at predicting the future. Okay, so the bottom line that in spite of countless differences, here the human brain and modern algorithms present some key elements of convergence. They have similar activations of representation. They share similar spatial and temporal hierarchies. And it seems that the similarity between deep nets and the brain is best explained by a simple loss function. And this resonates with a key idea in biology, the idea of convergence, where you can have two vastly different species that converge onto the same biological structure or the same functional apparatus. The classic example is the case of bats and birds, which have independently evolved in many ways very similar wings. Here it seems that deep nets and the brain, which are vastly different agents that are trained in vastly different ways, that have access to memory and energy in a vastly really incomparable ways, really. These two systems seems nevertheless to converge to partially similar representations, which suggests that a simple learning objective like the one that we use to train deep learning today is a sufficient rule to explain the rise of these complex and high-level representations. Okay, I'm not sure how much time I have left, so I can either conclude now or spend 3 more minutes on an extra experiment. So if I'm not being stopped, I'll just continue. Okay, so what I've shown you here is really the glass half full, because we simply observe a similarity here between language or speech models and the brain. And we just capitalize on this simple observation. It's just an empirical fact, in a sense, to observe this similarity. But we also know that deep learning algorithms are far from being as efficient as humans in many ways. And so the second part really is about the glass half empty, right? We know that these algorithms are not as good as the human brain, and therefore we know that something is missing from this big picture. So the goal for us is to try to come up with hypotheses that would improve the laws that we use to train these algorithms, to make them more brain-like and perhaps more efficient at learning intelligent functions, at integrating information in a more efficient manner. So one of the hypotheses that we are working with is the following. The standard way to train a deep net, at least in the context of language, is to try to predict the next word given the context. If you have once upon a, the algorithm is trained to predict the very next word time. What it is not trained to do is to predict what's going to come 2 words, 3 words, 4 words, 100 words from this context. So it's a very myopic, short-sighted objective. What we think the brain is doing is something which is not that myopic. Perhaps it tries to predict what the next word will be, maybe even the next phoneme and the next sound. But a critical idea in neuroscience is this idea that different levels of the hierarchy will be optimized or tuned to predict more distant futures. And so to try to test this hypothesis, we replicated the approach that I've shown you before, this time on 345 subjects that were curated by Nastas from Uri Hassan's lab. Each subject were listening to about 17 minutes of audio stories in the MRI scanner. And again, we first quantified the extent to which the representations of standard deep learning algorithms like GPT-2, something which is trained to predict the next word, would be with regards to the brain. And we observe a pretty similar type of brain map as I showed you before. Now, what we also tried to do is see whether we could improve this mapping between the deep net and the brain if we enhance the deep net with representations of the future, either predicted by the deep net or deep representation of the deep embedding of the future words. And what we observe is that we can improve this mapping, suggesting that we can identify representation in the brain which corresponds to these anticipatory anticipatory representations of what's going to come here in the human brain. Now, what is quite interesting is that the organization of these forecast representations of these predictions follows the expected hierarchy, namely the early auditory areas seems to forecast something nearby in the futures, whereas the prefrontal areas and the parietal areas seem to forecast representations which are much further away. And similarly, the early auditory areas seem to forecast predictions which are pretty shallow in terms of how deep they are with respect to the deep net, whereas the prefrontal areas and the parietal areas seem to forecast much more abstract, much more deep representations as indexed by DeepNet. So this suggests a simple idea. It suggests that the human brain here is not just trying to predict what the next word will be, it's trying to predict deep or latent representations of future words. And depending on where you're looking at in the hierarchy of the human brain, you can evidence different depths of prediction and different latency of these predictions. Okay, I will stop here, but before I take question, I would like to emphasize that this is a teamwork, and I'm really privileged to be working with a team of great students and engineers. The work that I've shown you today was done together with Charlotte Cocheteux and Juliette Millet, as well as Alexandre Grandfort, but the whole team really is moving on to progressing on these fronts. Thank you very much for your attention.
Thank you very much, Dr. King, for a fascinating lecture. I would ask everyone to hold their questions until the discussion period. And now I'd like to invite up Dr. Zirui Huang, who is an assistant professor at the University of Michigan, working in the Center for Consciousness Science, who's been doing some exciting neuroimaging work moving beyond spatial circuits into thinking about temporal circuits of functional brain networks. Dr. Huang.
Hi, good afternoon, everyone. I hope everybody is doing great and is enjoying the dry heat here. This is something special for people from Michigan. First of all, I would like to thank the kind invitation from the conference organization. It is my great pleasure to be here and to share and discuss my recent work about brain and consciousness. I'm truly honored to be on the same stage with so many outstanding scientists, either physically or virtually. Before giving an outline of my talk, I would like to start with some background introduction about my research field and by what means I study brain and consciousness. So I studied brain using functional magnetic resonance imaging, and what we get is the blood oxygen level dependent signals reflecting the neural activity. We have tens of thousands of signals from the voxels across the whole brain. Then the question is, what do we do about them? So one conventional way is to make correlations of these signals, which we call functional connectivity. It tells us to what extent a pair of brain region that is temporally coordinated. And there's a way to represent the functional connectivity relationship across regions, which is called functional connectivity matrix. And by thresholding the matrix, we could make network partitions. It tells us the brain regions within a given network are highly connected, then may serve particular functions such as visual, somatomotor, or attention-related process. Indeed, the neuroscientific study of consciousness versus unconsciousness regarding the functional connectivity has yielded important insights into the mechanism of consciousness. However, today, I will lead you to look at the data from different perspectives and hopefully provide some new insights about brain and consciousness. So let's go back to the brain signals. We could visualize them directly onto the brain without making correlations or time averaging. Then here, what we see is the dynamic brain activity recorded during resting state. So we could find that some brain regions are co-active at certain time points, and they resemble a predefined network. And sometimes the regions are globally co-active, and subsequently they are dissolved and form another network. So beyond viewing the brain network that is in a static sense, you could see some patterns that are recurring or alternating in a non-random way. So in addition, you may also have the intuition that dynamic brain activity are spatially organized in a delicate fashion, and you may ask if there's an overarching principle that regulates the dynamics. So therefore, in a broader sense, we are interested in the brain's temporal regularities and the spatial organization rules, and how they are related to consciousness, and how they are changed when consciousness is changed or diminished. So along the two lines of investigation in time and space, I will first talk about one of my recent studies that introduced the concept of temporal circuit of consciousness, which is followed by another study identifying a critical brain structure that regulates the temporal circuit and serves as a gate for conscious access of sensory information. So after that, I will discuss my ongoing work about brain's functional geometry towards a multidimensional account of consciousness, as well as the relationship between functional geometry and the temporal circuit. So let's start with the first one, temporal circuit. Human consciousness is thought to consist of both a person's self-awareness and awareness of their environment. So evidence from functional neuroimaging studies have reviewed two distinct cortical systems that support consciousness. So one is the default mode network, responsible for various kinds of interception, from mind wandering to recollection and planning, and the converging lines of evidence have showed that the default mode network underlies self-directed cognition. And activity in the default mode network is anti-correlated with the activity in the so-called dorsal attention network. So the more active one of the two networks is, the less active the other tends to be. And the dorsal attention networks becomes activated when a person is focused on a task. As we go about our waking lives, our stream of consciousness typically cycles through many such alternations between interception and outward attention throughout the day. And it appears that the back and forth dance between these inward and outward mental states may be fundamental to brain function. And studies have shown that the anti-correlation of the two networks is vital for maintaining ongoing interaction between the self and the environment that contributes to consciousness. However, there were a few problems. So first one, the evidence for anti-correlation had been determined by averaging images collected over several minutes. By such averaging, a data processing technique called global signal ration that reduced noise is often used, but global signal ration could falsely indicate existence of anti-correlation, which may be an artifact of data processing. Therefore, unequivocal evidence for default mode dorsal attention anti-correlation was lacking. Furthermore, even assuming anti-correlation, the dynamic relationship of the default mode network and dorsal attention network to other networks that is critical relevance to consciousness has not been investigated. So to fill this gap of knowledge, analysis of dynamic brain activity without using a global signal regression is necessary. Accordingly, we hypothesize that in the conscious brain, the dynamic switching of networks, including the dorsal attention and default mode network, occurs along a set of structured trajectories, which might be conceived as a temporal circuit. And that this temporal circuit is disrupted during diminished consciousness. To examine if our hypothesis could be held true across different types of reduced consciousness, we collected evidence in people rendered unresponsive, whether by anesthesia or a neuropathological condition, so including propofol and ketamine anesthesia, patients' diagnosis in minimally conscious state, and patients' diagnosis in unresponsive wakefulness syndrome or vegetative state. It consists of 4 resting state functional MR data set with conscious baseline intermediate states such as light sedation and unresponsive states. So here, rather than averaging activity over long periods, we wanted to investigate how moment-to-moment brain activation unfolds over time and to avoid the issue of global signal regression. We employed an unsupervised machine learning approach, K-means clustering algorithm, and looked for brain areas that are consistently activated together. And we classified fMRI volumes into K clusters based on their spatial similarity, and thus produce a set of co-activation patterns of the brain states. And we determined the optimized K that has 8 co-activation patterns by treating off the inter-data set similarity of the co-activation pattern distributions. But I will skip the technical details here. And here are the spatial maps of 8 co-activation patterns or caps. 2 of the caps correspond to the default mode network and the dorsal attention network, and the 6 are related to other known networks underlying brain function, including the frontal parietal network, sensory motor network, visual network, ventral attention network, and 2 networks representing cross-brain states of activation and deactivation. The 8 co-activation patterns can be divided into 4 mirror motifs with a strong spatial, let's see. Here, okay. With a strong negative spatial similarity. For instance, the default mode network was accompanied by co-deactivation of the dorsal attention network, and vice versa for the dorsal attention network. To illustrate these motifs, we could use the yin-yang bagua theory as an analogy. The theory consists of 8 symbols derived from yin and yang to represent the fundamental principles of reality. That is the energy balance to create matter. And I know this is metaphysical, yeah. But I should say that I use this analogy for entertainment purpose only, not a scientific interpretation. Let's go back to science. Having these co-activation patterns, we examine the differences in occurrence rates among different conditions. The occurrence rates of each co-activation patterns was quantified by dividing the number of fMRI volumes belonging to a given co-activation pattern by the total number of volumes per scan. And during unresponsive conditions, the occurrence rates of the default mode network and dorsal attention network were significantly reduced. And this phenomena was reproducible in 2 independent data sets of propofol anesthesia and generalizable to ketamine anesthesia and to neuropathological patients. We also found specific change of co-activation pattern occurrence rates during various unresponsive conditions. And comparing to conscious condition, the increased occurrence rates of visual network, ventral attention network was seen during propofol-induced unresponsiveness. And decreased occurrence rates of frontal parietal network, sensory motor network, and increased occurrence rate of global network co-activation, global network co-deactivation was observed during ketamine-induced unresponsiveness. And the neuropathological patients shared both effects with propofol and ketamine anesthesia. So together, a key finding is the suppression of antiphasic co-activation of the default mode and dorsal attention networks that occurred in various conditions of behavior unresponsiveness. So given that the result was obtained with different anesthetic agents with distinct molecular targets as well as in non-anesthetized neuropathological patients, so we suggest that co-activation patterns of default mode and dorsal attention networks are two fundamental signatures of consciousness. So in the next step, we wanted to delineate the temporal dynamics of these co-activation patterns. Let's start with an example. Here is a sequence of co-activation patterns consists of A, B, and C. Transition probability can be described by a Markov process, where the probability of A at time t+1 is determined by A itself or B or C at time t. And these probabilities were encoded in a transition probability matrix. So we refer to the undiagonal entries in the full transition probability matrix as the persistent probability. That's the probability of remaining in a given co-activation pattern. And we refer to the off-diagonal entries in the diagonal-free transition probability matrix as the transition probabilities. That is the probability of transitioning between two distinct co-activation patterns. So using statistical analysis such as permutation tests, we could address that. First, given a current state is A, whether it prefers to stay at A. And second, given the current state is A, and given it is moving out from A, which are the preferred passes or to other states. So here is a video, for example, showing the dynamic transitions between co-activation patterns in conscious wakefulness, propofol-induced unresponsiveness, ketamine-induced unresponsiveness, and patients with unresponsive wakefulness syndrome. So this is just give you a very intuitive sense on how brain state transitions look like. If you look at this video for a longer time, you may observe some regularities. And here's a summary of the results. So we observed distinct temporal dynamics across different conditions. So the gray arrows indicate significant preferred passes and non-preferred passes, and the red and green arrows indicate significant higher and lower probabilities comparing to baseline conscious condition. So the take-home message is that during conscious condition, the co-activation patterns follow structured transition trajectories with balanced preferred and non-preferred pathways. In contrast, during unresponsive conditions, there are less trajectories reaching default mode and dorsal attention network, and the trajectory space is monopolized by a few attractors such as visual network and ventral attention network. So in other words, in subjects who are either under anesthesia or unresponsive due to neuropathological conditions, both default mode and dorsal attention networks were visited less often, meaning that the number of times they were activated within the cycling patterns of networks was reduced. And these observations were supported by examining the entropy of Markov trajectories. So this approach measured the descriptive complexity of trajectories between each pair of co-activation patterns. A lower descriptive complexity from a starting point to its destination indicates a higher accessibility for the destination. And the accessibility of the co-activation pattern is defined as the inverse of descriptive complexity, and the node size is proportional to the in-degree accessibility. And we could see that conscious brain activity is characterized by a set of structured dynamic transition trajectories in which the accessibility of distinct brain states is relatively balanced. And the key finding across these various conditions is that unresponsiveness was associated with isolation of default mode and dorsal attention network from the trajectory space. That is common to any form of diminished consciousness. And we conclude that human consciousness relies on a specific temporal circuit of dynamic brain activity characterized by balanced reciprocal accessibility of functional brain states. And the disruption of this temporal circuit exhibiting limited access to default mode and dorsal attention networks appears to be a common signature of unresponsiveness of diverse etiologies. Okay, I will move on and talk about which brain region regulates the temporal circuit that also serve as a gate for conscious access of sensory information. We hypothesize that there exists a critical brain structure between primary sensory and the association cortices that regulates brain network transitions associated with conscious access. So here we refer to conscious access as the availability of consciously perceived information to cognitive processors such as those that mediate functions like working memory, verbal report, or motor behavior. So in this work, we performed functional MRI in two experimental paradigms. So in the first experiment, we applied the general anesthetic propofol as a tool to modulate the level of consciousness of healthy volunteers who were instructed to engage in mental imagery tasks. We aim to determine which brain region disables the brain network transitions, rendering a failure of conscious access. And in the second experiment, we evaluate the identified brain area in a psychological setting where the content of consciousness was manipulated in a classical backward masking paradigm in awake, healthy participants. And here we ask whether the spontaneous activity of the brain, the same brain area, was predictive of future conscious access. And we report both sets of experiments supports a key role of the anterior insular cortex in regulating brain network transitions and gating conscious access. So here is an overview of the study design. Healthy volunteers were studied using functional MRI during and after intravenous propofol infusion, and the infusion rate was adjusted to achieve stepwise increasing target effect site concentrations and indicate, as indicated by the green curve. After that, the infusion was terminated to allow for spontaneous recovery, and behavior responsiveness was assessed by motor response, that is squeezing a rubber ball by hand, which defined the periods during which participants retain behavior responsiveness, pre-IOR, loss of responsiveness, IOR, and recovery of responsiveness, ROR. And two resting state baseline and two task baseline recordings were done before and after propofol infusion. So between the motor response trials, participants were instructed to perform mental imagery tasks such as playing tennis, navigating at home, or imagining of squeezing the rubber ball but not actually squeeze it. And let's look at the results. So during the baseline condition, mental imagery and motor response tasks produce distinguishable patterns of activation in specific regions of the brain. So the signature brain regions for the tasks are the supplementary motor area during tennis imagery, the parahypoplace area during navigation imagery, and the supplementary motor area during squeeze imagery, and the primary motor cortex during motor response. And verbal instructions elicit widespread cortical and subcortical activations, including thalamus, auditory cortex, anterior insular cortex, and dorsolateral prefrontal cortex. And during propofol infusion prior to loss of responsiveness, activations were mostly preserved. And during loss of responsiveness, mental imagery-related activation were absent, where verbal instruction-evoked activations were attenuated and constrained within the thalamus and auditory cortex. And instruction-evoked deactivations in bilateral anterior insular cortex were observed. After recovery of consciousness, activations evoked by motor response and verbal instructions were recovered, whereas mental imagery-related activation were less prominent. And during the second baseline, base 2, the patterns of activation in different tasks mostly mirror those seen during the first baseline recordings. So to this end, the results are in line with the notion that sensory inputs can be received but not perceived during anesthesia, because we see a preservation of primary sensory processing but a disruption of high-order association cortices. Next, we sought to determine the processing stage along the cognitive hierarchy at which the task-induced brain activity breaks down during loss of responsiveness. We include a bunch of brain regions of interest that are involved in the tasks, and we classify these regions along the cognitive hierarchy based on cortical gradients. I will explain this method in detail later on. What we need to know here is that there is a primary cortical gradient from unimodal to transmodal areas, which is thought to guide the information, the flow of the information across the cortex, allowing sensory signals to become increasingly bound to non-sensory information and transform into more abstract contents. And here are the functional networks reordered along such unimodal to transmodal axis. We then localize the brain regions within these networks. We see that the ventral attention network situated at the intermediate position of the cortical hierarchy, and the anterior insular cortex identified in our data was exactly in the ventral attention network. We next test our hypothesis that whether the anterior insular cortex plays a role in mediating network transitions. So for that, we first tracked the time-locked brain network transitions required for conscious processing, and they used this to identify the critical brain region that mediates such transitions. We found that during conscious conditions, there was significant positive modulation in the dorsal attention network and a significant negative modulation in the default mode network for all imagery tasks. This was consistent with the known default mode dorsal attention network switch when participants engaged in a cognitive task. So confirming our predictions, the default mode dorsal attention network switch was abolished during loss of response period, where you could see that the blue curves are almost flat. And next, we performed intersubject correlation analysis between instruction-evoked activation in the anterior insular cortex and the dorsal attention and default mode network modulation index. So supporting our hypothesis, a higher anterior insular cortex activity following the verbal instruction was associated with a larger gain of network transition during a sustained mental imagery task. So taken together, the results suggest a primary role of the anterior insular cortex in mediating the network switching necessary for conscious axis. And finally, we test the causal roles of anterior insular cortex in gating conscious axis in another set of experiment under a psychological setting. So here, participants were briefly shown either face or scrambled face image, followed by a high contrast image using classical backward masking paradigm. And we contrast the stimulus-induced activity of seen versus unseen face in a near threshold condition. We find that higher stimulus-induced activity in the anterior insular cortex, dorsolateral prefrontal cortex, and anterior cingulate cortex and thalamus was associated with the perceptual awareness towards seeing a face. And importantly, the perceptual bias also occurred at the stimulus onset for these regions. So these effects at the stimulus onset could only be explained by pre-stimulus rather than post-stimulus activity, considering the hemodynamic response delay of fMRI signal, and thus indicate a causal impact of pre-stimulus activity on the subsequent perceptual awareness. And to this end, the anterior insular cortex showing deactivations during loss of consciousness in the pharmacological experiment coincide nicely with the anterior insular cortex identified during the perceptual awareness in the psychological experiments. And people would wonder why anterior insular cortex plays such a unique role. And here, as I have discussed, anterior insular cortex is a central component of the brain's ventral attention and the salience network, which is situated at the intermediate position between unimodal and transmodal areas along the brain's functional hierarchy. Anatomically, the anterior insular cortex is composed of unique clusters of large spindle-shaped pyramidal neurons in layer 5 called Van Economo neurons. And these neurons establish long-distance fast relay of information throughout the cortex. And functionally, the anterior insular cortex has been recognized as a region playing a broad range of roles across many different domains. The key role of the anterior insular cortex seems to be identifying and prioritizing salient stimuli in the stream of continuous sensory information and sending signals to the system responsible for allocation of top-down attentional resource to the relevant sensory representations. And to conclude, the results suggest that this function of the anterior insular cortex disables brain network transitions. Pre-stimulus activity has an impact as well. And the anterior insular may be a gate for conscious access of sensory information. And this cortical gate occupies an intermediate position along the neurocognitive hierarchy. And to this end, I have discussed the temporal circuit and its modulation, and I will just briefly talk about one of our recent work about functional geometry and its relevance in supporting a multidimensional account of consciousness. As introduced by Christoph Koch, Professor Christoph Koch this afternoon, consciousness is a multifaceted phenomena that can be altered by various causes. And conventionally, consciousness is defined by two separate components, awareness versus wakefulness or vigilance. So although this definition is useful heuristically, it's questionably assumed that normal consciousness is associated with the highest level of awareness and wakefulness. For altered state of consciousness, such as those induced by psychedelic drugs or psychiatric causes, such as schizophrenia, are difficult to fit into such scheme. And these individuals can be fully aware and awake with even richer conscious experience than in a normal conscious state. And recent years, multidimensional representations of consciousness has been proposed from a theoretical perspective. And however, an important knowledge gap has been the identification of what these dimensions are in neural terms and how they are associated with the brain's neurofunctional properties. And we address this gap of knowledge by developing a brain-based account of consciousness as multidimensional phenomena. And we hypothesize that the state of consciousness can be defined by multiple neurofunctional dimensions of the brain. And thus, a change of conscious state is fundamentally the manifestation of a change along one or more of the neurofunctional dimensions. So here, instead of looking for discrete brain regions, we are more focused on the brain's intrinsic functional geometry. A recent advance in neuroimaging offers approach to test this in the context of consciousness, and the cortical gradients, which subsumes multi-functions and functional networks along a continuum, are attractive candidates for neurofunctional dimensions of consciousness. And the gradient range from unimodal systems that underpin perception and action to the association cortex implicated in abstract cognitive functions. So to illustrate this concept using the analogy of land, one could define a region by absolute Cartesian coordinates, or we can measure the slope of elevation or spatial change in vegetation types. So assessing the rule of topographical continuum is the basis of cortical gradient mapping. So I will skip the technical details of the gradient analysis. It just gives you a very intuitive sense that after dimensionality reduction, we have three major cortical gradients along unimodal to transmodal, and the second one is visual to somatomotor, and the third one is from visual default mode to other areas implicated in multi-dimension. And we can plot in 2D or 3D dimensions. And we hypothesize that different neurofunctional dimensions of consciousness can be represented by different cortical gradients. And the disruption of the consciousness is associated with a degradation of one or more of the major cortical gradients. And we test the hypothesis by using a couple of data sets, including propofol sedation, propofol anesthesia, ketamine, unresponsive wakefulness syndrome, and psychiatric patients. And we classify them as depressed state of consciousness and the conscious with psychiatric diagnosis. And they have distinct features in terms of consciousness. So for example, propofol deep sedation lose the awareness, and ketamine anesthesia affect sensory organization. That's same with the psychiatry schizophrenia patients. And for general anesthesia, the arousability is lost, so cannot be aroused by painful stimulation anymore. And we can visualize those gradients in 2D or 3D gradients, and we can measure quantify the numerical range of each gradient and compare them between different conditions. And we found that the gradient 1 was degraded in those conditions, indicating loss of awareness. And the gradient 2 is lost when sensory organization is disrupted. And finally, gradient 3 is degraded when subject was no longer arousable. And patients with bipolar disorder, ADHD, did not show such major changes. So together, the results shows that the gradients can be a candidate to represent multi-dimension of consciousness. And here we only focus the first 3 gradients. The optimum number of gradients need to be determined. And I will skip that functional geometry and the relationship of temporal circuit. Just to show you the main point is that we can convert gradient into a 3-dimensional space and localize those networks and correlate those networks with the temporal dynamics. And we find that they can explain to each other. And it seems that the temporal circuit is somehow constrained by the functional geometry. And I think that's all. So I start with the 2 networks and the gating rule of the ventral attention and the sensory motor and the conscious axis, temporal circuit, 3 dimensions, and one remaining question is that how subcortical regions is associated with temporal circuit. That warrants future investigations. So finally, I would like to thank to my colleagues who are actively involved in this study and the general support from NIH, and the special thanks to my mentors, Professor Hudas and Professor Mashour, for the excellent guidance and great support, both personally and scientifically. Thank you.
Thank you very much, Dr. Huang, for that fascinating talk. We are now going to move on to our third speaker, Dr. Anirban Bandyopadhyay, who is coming to us from Tsukuba, Japan. He is a member of the Nanocharacterization Unit of the National Institute for Material Science, and he's going to be giving us a very different perspective of connectivity at a different scale. Anirban?
So, first of all, I would like to thank everybody for joining this TSC, and I would like to thank Stuart for inviting me to give this presentation. And it is every year it is a great pleasure for me to come and present the recent developments that we have made. And I would also like to thank AB for holding the main structure at the background and running everything smoothly. Nothing goes wrong if AB is present over there. So with further ado, I would like to start my presentation. So today I'm going to talk about only about neuron studies that we have been studying for the last 12 years, and we have been publishing regularly. More than 8 to 10 papers are already published, and many more are on the way this year in press, or it will come out very soon. So you can get all our papers in ResearchGate, because my student Pushpendra and Komal, they are very active in uploading the papers in ResearchGate. So you don't have to purchase. You can just download freely from there. And so my first presentation today. So this is the device that we use for measuring cultured neuron. So primarily what we do is we have a coaxial probe. The coaxial probe is also designed here. It almost, since I'm in the laboratory, I can show you. So this is almost, this is you can see the tiny gold colored tip. So these are the tiny tips, extremely expensive, because the gold, it goes to the nanoscale, the tip, and inside that there is a glass tube, and inside that there is a platinum tube. So platinum tube is at the core, and on top of that there is a glass, and then there is a gold. So we can insert deep inside a neuron or on the top of the neuron on the membrane, and we can measure the electrical communication there. So we have many such probes. So inside one neuron, we can put many such probes, and then simultaneously we study what is the electromagnetic or electrical communication through the neuron membrane or inside the neuron cell through the microfilaments and neurofilaments. And so this is basically the design, and this is a scanning electron micrograph and helium ion microscope. We observe it. So it is at the very edge. You can see platinum and gold. So inside the scanning tunneling microscope, we can put the cultured neuron plate, on HOPG or other kind of surfaces. And then there is a piezomotor which has atomic scale precision. It moves and then take the image. This is kind of one of the images in normal microscope with our coaxial probe, and that is inserted inside the axon, and we do this kind of characterization. So this is the published paper from 2016, Journal of Integrated Neuroscience, and so we have this coaxial probe, and we can insert the electrodes in any of the any point, and nerve spike is a circular in shape, and we can measure how the communications are happening to the boundary, and at the same time we can also artificially create microtubules, neurofilaments, and beta spectrin and actin filament structures which are periodically on top of it. So we get the electromagnetic simulations by solving the Maxwell's equations. That almost looks like this. And we carried out studies on tubulin protein, actin, beta spectrin, ankyrin, and several different kind of associated proteins inside the neuron and neural structures, synaptic junctions, like clathrins, and several different kind of proteins we studied, and we collected a database of electromagnetic resonance spectrum. But the electromagnetic resonance measurements started in the 1930s, in 1928 and the 1930. First measurements were done, but later in the 1960s, electromagnetic resonance measurements were, if you put inside a cavity in the solution and you study. For us, what we do is we have protein molecule or protein complex molecules, but nearly dried. It is wet, but not too much water inside. Because if too much water and we connect the probe, then water conducts, and then communication does not happen through the biomaterial. That's a huge challenge. So many of the papers that you will see published are in solution. They put protein and then they put the electrode. All these measurements are measurement of water, not the measurement of protein molecules. So you see that in their results, you will see that when they show the peak, you will see with solution, without the sample, and with the sample, you'll see the pattern looks similar. The peak distribution, ratio of the peaks, all are similar. Little bit of intensity change because they add impurity. And the protein works as impurity. So all those measurements are non-reliable, very wrong measurement. So we have to get rid of water, because we get rid of water completely. So in our paper in Journal of Integrated Neuroscience, we have spent a lot of space. It is a 60-page paper, but you will find that 10 to 15 pages we have spent on how to get the water out. If you take the too much water out, then data is wrong. And if you take a little bit of water and too much water, then also it is wrong. So you have to control it with a particular prescription we have given, and you can refer to the papers. So when you insert the electrode deep inside, you penetrate through the membrane, that is the cover of the cell, the skin of the cell. And when you go inside and you switch on, if the electromagnetic frequency is on. So for each protein and protein structures, they have a particular kind of frequencies. So when you go inside, see here that you get different signatures of electromagnetic frequencies, and you finally encounter the microtubule bundle. So it is not a smooth journey. There are many different kind of steps, and you repeatedly perform the experiments several times to understand that it is a general procedure. So all these technical background which are necessary to study the microtubules deep inside a neuron, all those were published in our first two papers in 2016. That's a 60-page long paper, and where all these technical details you can find. So many different fundamental things we need to take care of, because vibrations are too much. There should not be much vibration, and how do you control the vibration with a piezoelectric setup, how cellular vibration, solution vibration, and how continuously we have to monitor the electromagnetic resonance frequencies of the environment, so to check that we are really measuring the material that we want to measure. So these are the kind of protections, and finally you get results like this. So this is published in last year. We published absolutely purified results. So after 6 years of the first paper, we published pure signals that you can see at different parts. So we put many coaxial probes at a different part of the neuron, and then we get the signal. So we find that when there is a firing, much before that, there are many events that are taking place deep inside the neuron. And in the filamentary structure, there are many order structures, not just microtubule or neurofilament. There are actin and beta spectrum, beautiful grid-like architecture just beneath the membrane. So all these are working together in a harmony, different time domain. They are communicating signals. So it's a whole new world that is hidden inside and which were unexplored, thanks to the work of Stuart and his lifelong passion and dedication, that several people got interested, like me, who got interested and started measuring this kind of data and documenting it. So now we have created a database, www.brainrhythm.org, and it is a database between NIMS, Japan, and University at Buffalo, UB, and we are giving this data free to the people to download and to advance the work. And you will get the raw data that we measure, and then several background information, phase correlation, and other kind of data which we do not publish in the paper, which are not useful for any paper publication. But if any researcher want to seriously do this kind of work, they just look at this particular figure. They cannot understand what is going on, because when you put many different kind of probes, you need to phase correlate all the probes. What are the phase relationship? Are they phase locked? So phase locking is with one system or two systems fine, but when you have say 6, 7, or 8 electrodes, and they have each pair has a gold and platinum. So say 8 means 8 into 2, 16, and they need to be correlated. Phase coupling needs to be there, and phase adjustment needs to be there to understand the magnitude or the changes in the peak shifts and other kind of details. So we since we have atomic resolution, we can identify the ion channels. Single ion channels has been studied very recently. I don't believe that Sheikman Nair's work, for which they were given the Nobel Prize, that they measured single ion channel. They claimed it, but I don't think so. They didn't realize it. It happened much later in around 2008, 2010. Several groups came up when the technology was much more advanced. Only then single ion channel was measured, imaged, and they successfully able to track down. And we have also tracked down, but we are not the first. We are not even the second. We may be 5th or 6th group in the world to see this. But what is our advantage is our resolution is similar to the other groups who have now atomic resolution sensors. But at the same time, we can image it also, the ions coming out. So this is an advantage. So simultaneously, both the things we can do. And so what we can do is we can pump electromagnetic wave. We can stop neuron to fire, and we can trigger neuron firing by suitable frequencies, that is the resonance frequency of the microtubule or resonance frequency of the beta spectrin or actin channel. So there are different kind of frequencies by which we can shut off. So the figure A you can see this is Journal of Integrated Neuroscience, and this is 2021, I think it was published. We go to now Journal of Neurophysiology paper, where we have shown a very sophisticated scanning dielectric microscopy. Scanning dielectric microscopy, for some reason, it is not popular in the West. I don't know why. It was discovered in 1995, in Japan, I think Tohoku University. They discovered, but we advanced it and made it suitable for neuron measurements simultaneously, multiple. So they scan it and pump the electromagnetic field to the sample and reflected and transmitted beams that is coming out. Reflection and transmission coefficients are measured. So using that, they image the structure. So we have been publishing several kind of papers, and this is one of the schematic. Neuron is here. So coaxial probe array is at the bottom and at the top, and it vibrate, and then we are able to get very high resolution, a fractal resolution image of the structure. But very high resolution means still say 60, 70, 80 nanometers or 100 nanometers, not better than that. So please understand that for cultured neuron, it's very difficult to get atomic precision. If we can do that, that will be the best. But still, we have not achieved it. And what happens is we get a similar kind of data, and then we apply diffuse spectrum, and then we get the actual image that we present. So it is very pixelate form, but with artificial using the Gaussian filtering, we show that as if we have imaged the real neuron, but it is very pixelate one in reality. So in the paper, when I show the video, it looks very beautiful, but actually in the paper, we have shown what you actually see. This is the kind of image that you actually see, and this is the kind of video that we show you. So there is no need to be frustrated if you are trying to do this kind of measurement for the first time and we end up with a very low resolution image. Actually, the images are very low resolution. So, so this is from the manuscript. So what we do is a neuron, how it is electromagnetically communicating with different neighboring neurons, and what are the frequency band it is using to communicate wirelessly with a different neuron. Wireless communication between different neurons was first published in 1928 by Ecole from Germany. But the point is that for some reason it never entered into became a part of mainstream neuroscience. We repeated the work, but of course, since this is it was 2013, 2014, so technology was much more advanced than 1928, and so we could image. And you see this beautiful video I've shown in TSC conferences many times. You can go to our publication in Journal of Neurophysiology, and you can see this image. And the video is also there online. So basically, what happens is millisecond time domain. It is known for the last 100 years in mainstream neuroscience that the millisecond ion communication happens. But of course, there is in the megahertz domain, in microtubule and other filaments, they do dipolar oscillations, and they also communicate. But the beauty is that because it's electromagnetic, so wireless communication is possible. So for them, membrane and other things don't exist. Only those vibrating in the same time domain, they only exist. So it is just like another neuron is just like another energy orbital, and as if a single atom is transiting between two atomic orbitals. Analogous. This is not real, but analogous. I'm trying to create a metaphor. How do we understand it? So what we find that we summarized in our paper, in our Journal of Neurophysiology paper, is that if you have many different kind of gap junctions, so electromagnetic field in a megahertz is a long range field. So what happens is it is able to see where the energy gaps are there, and then the whole system as a whole try to regenerate the symmetry by closing the gap, opening the gap, regenerating the gap, building the gap. And that's how the firing starts. So there is a close communication between ionic firing and dipolar firing. So these two kinds of firing work side by side and complementary to each other. And dipolar firing is much, what should I say, is much faster, is 200 microseconds faster than the millisecond firing that we see in the neuronal domain. So there is a time to change the view. So this is the work. I would like, so with a scanning dielectric microscope, by tuning the frequencies, you can image either the membrane, the beta spectrin just below the membrane, or the microtubular neurofilament bundles. So megahertz, and then several hundreds of kilohertz, and then with 1 to 10 kilohertz, then you can see the membrane. So which frequencies you are using to image a particular biomaterial, depending on that, those kinds of dipoles will be visible to you. It's a simple dielectric property. This is not an incredible science we are doing here. Only thing is that nobody tried to see or perform these simple experiments, which technology were there for which in the 1970s, 1980s. And of course, in 1995, since 1995, instruments are available. So let us summarize what we did. So 1907, Lapik came, and the Lapik from, I think from France, he first suggested that, look, neuron ions come through membrane, and then they come to the soma, and then it bursts, electrical bursts happen, which still read in the textbook, and then circular potential is generated, and that passes to another. In 2016, what we said is deep inside the membrane, there are neurofilaments, and then they can accept the quanta or electromagnetic noise or something. But since these are ordered structures, filaments, noise converts into a meaningful signal. And as it passes back and forth through the actions and then the neural branches, it gets ordered. And a pair of cylindrical electrical ring is generated. And these rings get activated one by one after another. And in between these two rings of field, the ion channels emits. So in the circular region, all the ion channels are activated at once. So it's a beautiful, meaningful signal. So if microtubules are melted, all the filaments are melted, will the neuron fire? Of course, the ion channel will fire, but that will not be able to control the time gap precisely. So to keep the millisecond time gap precise, the human brain consciousness or cognition is attributed to the precise control of the time gap between spikes, not just making the spike is not enough. So that control is actually provided by by many ordered structures. And I must say that there are many people who contributed much before our work, like 2013, Zhu et al. From Harvard University. They first said there is an ordered crystalline structure just beneath the membrane. And Hirokawa-san, for 30 years of his life, he worked on microtubule and filamentary bundle to suggest that inside the neuron, it is not a jelly. It is a very ordered, unidirectional architecture of microtubules, and gap between them is only 50 nanometer. So it is highly and very, very strong ordered unipolar structure throughout. So this is, so our work, as you can see that when you see these great works, our work is just a little extension of the previous work, and we have tried to put together many different advancements which was lost in history, and great people, they dedicated their life to build these technologies and build this understanding. We have integrated them together and created a beautiful presentable form, so that that we are presenting in front of you. And you can see that already the discoveries which were there, if we just put them together and arrange it in a proper manner, it gives a beautiful integrated picture of the whole neural network system, which redefines our idea about the neural network, human brain, and its cognition. So then finally, in the last 6-7 years, we understood that and that one thing that we have to do very clearly, that is just like in the normal quantum processes, when we study the normal quantum processes, we go to the cryogenic system, we cool down the temperature, and then we try to get the quantum properties out. Biological system, even if there are all the molecules in our body are quantum mechanical system, right? Each molecule is a quantum mechanical system. They are working in room temperature, and they are, of course, they have the quantum mechanical property. So taking one molecule or the complex molecular system and exhibiting their quantum properties is not that breakthrough, which would be if we can show that biological structures, biological properties have the reflection of the quantum phenomenon. Whether that kind of rigorous study, if we want to perform, we need to change the very way we are looking into the biological system. Many of the biological structural works that people try to implement with quantum communication and quantum properties, they take the material, cool it, and try to observe and the quantum properties. I think that is a very, very wrong idea. What we have to do is to apply microwave or kilohertz, megahertz, gigahertz, terahertz, different kind of frequencies, and then try to see with light, polarized light, how that material is behaving. Then input is wireless, output is wireless. And another beautiful thing that we have argued in paper after papers in the last 6-7 years, we have at least 8 to 10 papers where we have argued that the idea of understanding a biological system, that we pump electromagnetic signal at say 1 to 10 megahertz, and output we are seeing 1 to 10 megahertz, what is happening out there? No, that's not the right way of doing it. Right way of doing it would be, say you apply gigahertz and you go to the kilohertz or megahertz domain, 10 to the power 6 times lower frequency domain, and try to see the beautiful pattern of vibrations generated over there. So those are not the direct hysteresis effect of the signal that you are pumping. For an example, you are throwing a big stone towards me and I scream, and that is not my natural expression. I scream because of the pain that I have. And then you model my screaming with different, if I throw this much force, Anirban screams like that. If I'm this much force, and you make a plot and you publish it in a Nature that this is the natural response of Anirban. This is the expression of conscious behavior of a human being. So that level of stupidity is when we pump energy and try to do. So what we do is electromagnetic, say microwave, and then 10 to the power 15 hertz laser, polarized laser come. Terahertz, and then we go to the hertz. So 10 to the power 6 order gap we keep, and we try to see the meaningful signal. So this is from Symmetry paper, journal. Last year, this paper was published. So what we did is, for long, I have changed. If you see this is my laboratory around, and if you see all around, you will find all electromagnetic frequencies up to 200, 300 megahertz, and then 10 gigahertz, and even to the terahertz domain. Several sources are there. And then laser sources are there for all the lights. And then we have optical bench, quantum entanglement, 16-channel quantum entanglement measurement systems are there. So many, so we pump the laser light, polarized laser, to the neuron. And what we do is we measure the optical ring, electromagnetic fields, which passes through, resonates a particular spectrum, and then we observe the light. So light is, if you send a polarized light, it rotates, it starts rotating, it creates, it takes a spiral path. So when it is projected, we see a ring. It's called optical vortex. So with optical vortex, you can read our paper, book published in 2020, where and in 2016, we have filed a patent of geometric musical language. So we say that the language of the biological system is not electrical signal or something. In the electrical signal, you can't find it. You cannot find it in optical signal. You cannot find it in mechanical signal. You will find it in the vortices. Vortices are circular or spiral field that is generated by the biomaterials. And we understood it in 2013-14, and we filed a patent, and a series of patents we have filed for the true biological language and then integration principles. So this is the experimental setup. So we pump the laser and we see light rings or magnetic field rings of magnetic field, rings of electric field, and mechanical vortex. That is almost like a sound or ultrasound. So 3 of the signals, vortices, come out simultaneously from the biological material, and we try to measure that. So you can see here, red line goes through and then green line goes through. So magnetic, and then you get the electrical, the optical light ring. So how does it look like? So, so the reviewer of this paper, they asked us to deconvolute all the, because when we pumped the neuron, we saw electric vortices, assembly of vortices, something like that. And they asked us to deconvolute, find out for each component, which are those. Because if you do not deconvolute it, we cannot start a true scientific debate and discussion that what could be the language of the brain. So all these data are now being uploaded in the www.brainrhythm.org. So in that database, we are giving away there's structures, files, softwares for analysis. So all the research materials that you need, it will be there. And so we deconvolute it. Deconvolution means we electromagnetically we resonate particular component and see which turns brighter, which one of here turns brighter or goes silent, which are the light rings, and then we identify, okay, this component may be responsible. So it's a kind of indirect proof that what could be the signal of the pure material. Pure material we can directly study and also get the optical vortex signal. So that is kind of these videos of electromagnetic resonant oscillations with quantum tunneling images I have shown many times. This is one of the setup where we measure the quantum entanglement. So you can see here is the oscilloscope, and we actually pump with a monochromatic laser. We split it into 2 parts using dichrosine of a BBQ crystal, and then 2 entangled photons are sent to form the different microtubules, and then we let it superpose, and we measure the signal, and if they are synchronously they are creating only one peak, then we understand they are entangled. If they are isolated, then they are not. So what we do, we apply the electromagnetic field. So microtubule is not, so these are the red one are the optical vortices, and then green one are magnetic vortices. So we studied similarly looking helical carbon nanotube to find out what kind of vortices come out of these biomaterials. And this is the quantum entanglement method. So here, if you look at both the channel 1 and channel 2, so we found that if you look at the, this is a complex plot, I understand, but there is an easy way to understand it. So you look at the projection below and you look at the projection below. You will find there is a similarity between the two, so channel 1 and channel 2. So 2 different of them. And then at very particular megahertz frequencies and gigahertz frequencies, we found that in the gigahertz frequency range, the 2 different kind of microtubules at certain frequencies, they remain entangled, but not always. At a very, very particular frequencies, it is there. So, so what we did is we took a tubulin protein. So this is a live blinking of the tubulin proteins. Me and my lab mates were talking. So these, you see parallel lines. Why do you see, because when laser light passes through tubulin proteins, they are because of the helices. 2 different sources are created because of birefringence, and they interfere. So for each tubulin, we'll find the parallel lines. They are the interference beams. So that is how it is generated. And so finally, I would like to thank everybody for being patient and listening to our work. And I finish today's presentation by showing you that at the different resonance frequencies, if we measure the quantum tunneling, at gigahertz range, we can find out, we can image the water channel, what it is doing deep inside microtubule. So the direct resonance microscopy is a beautiful instrument that without touching it, even if it is a giant structure, doesn't matter. If it is a big biological structure, taking small part of it, and then you can image that particular part without touching it from far, because everything will be resonating, and only that part energy will be concentrated, and that will affect the whole scenario. So thank you very much. And at the end of this presentation, I would also like to thank once again organizers, and especially Stuart, for giving me this invitation and presenting our work. And thanks to Abby for providing all the kind of background help, as you have always done this. And I would also like to say that all the participants of TSC 2022 that I'm missing all of you. It would have been great if I could go there, but because of the corona, as you know, that if I come back, I will be put into quarantine, and then that is a very pathetic experience. So I wanted to avoid that, but definitely next year we will be meeting, and till then, goodbye and enjoy your time. Thank you.
Thank you very much, Anirban, for a fascinating talk, and I'd like to invite Zirui up to the stage. Dr. King is not able to join us for the discussion, and if anybody has questions, please line up at the microphone and kindly keep your questions focused so we can hear as many voices as possible.
Can I have a question for Anirban?
Yes, of course.
Hello, this is a question for Anirban. It's an honor to be able to ask you this. So, from my understanding, your work describes, well, first of all, this question is on the topic of beat frequencies as it relates to transcendence. So bear with me here. But your work describes a bottom-up cascading process of resonant beat frequencies whereby consciousness potentially operates from the microscopic quantum level up into the macroscopic regime of space-time geometry measured in EEG. Do you think it's possible for consciousness to utilize the same dynamics to operate in a reverse top-down way, whereby, say, someone listening to binaural beats could entrain their macroscopic brainwaves to resonate with quantum oscillations, effectively shifting the seat of their awareness to the very fabric of the universe? And I guess of note is in a lot of accounts of people who've reached transcendent states, there's always a sense of this vibrational state, and a lot of times it's measured in a coherent 4 Hertz frequency band. So I'm just wondering if there might be some correlation between this and, well, I don't know, figure it out.
Yeah, I got it. Are you able to hear me, George?
Yes, we can. Perfectly.
Yeah. So in 2014, we published that there is a triplet of triplet resonance frequencies. If you start measuring from the alpha helices, single protein scale, neuron scale, microtubule scale, neuron scale, and then cortical column, up to cortical column, and the bundle of neural network we measured. And we showed that triplet of triplet, there is a hierarchical integration of frequencies in a geometric way. Just my previous speaker was showing very exciting plots in the end of his talk. So those kind of plots we have been doing for a long time, but in the molecular scale and that region scale. So I was very fascinated seeing that presentation. So you are absolutely right that there is a hierarchical integration and goes to the top. But can we apply certain frequencies in the brain and to go all the way down or all the way up? Because it could be that we are not end. Maybe all the humans are also connected with certain frequencies because circadian rhythm is there. So it could be the planetary systems are also connected by the frequencies. So I would like to tell 2 results that we are getting. 2 results that we have. One is Professor Anirban Datta from University at Buffalo. He was measuring in the Petri dish organoid brain, and he was measuring the electric signal, and he found triplet of triplet. And then he searched in Google, and then he contacted me that Anirban, I have seen it. So and another person from United States, he put in his brain a function, a spectrum analyzer. You can purchase it in $20, $30 in Amazon. Purchase it with a cable, put it in your brain, and you will find at 6 to 21 megahertz. Exactly, we published in 2013 Applied Physics Letters, the triplet of triplet band for microtubule. You will find similarly on your brain head. So you just touch it and see the magic. So it's a beautiful triplet of triplet symmetries everywhere. Still, I am enjoying the beauty and still I am learning. So I understand your question, but I do not think that we can pass ethical clearance. But next month I'm going to study lots of human subjects, but we will be only measuring the signals coming out. So triplet of triplet signals are everywhere. I think, I don't know, my knowledge is limited, but I am seeing everywhere it is coming up. So that's all I would like to say, because there are lots of questions I can see here. So we should take one by one.
Thank
Over
you,
to John.
Anirban. Next question, please.
Hi, thank you to Dr. Huang. So for just the part of the face detection experiment,
Yep.
I was just wondering, was it different faces and did they have an emotional component? And I was wondering, because it looks in your results like there is an asymmetry that we observe with emotional valence and motivation with interhemispheric asymmetry in activation. And that would involve limbic system structures like the insula. And so, first, was there an emotional component to the images?
That's the neutral phase, only one picture.
Okay.
Throughout the entire experiment.
Okay, cool.
And repeat many times.
Wow.
Yep.
And I noticed you mentioned a difference at time 0, I believe. And so how do you explain that? Could it be because of filtering, group delay, or like a methodological
Good question.
thing?
So no filtering was applied in the data. It's just raw data, then make the time average, then a time log to the onset. And the onset difference for the two seen, unseen phase is due to the spontaneous pre-stimulus spontaneous activity.
There was no cue, right?
No cue.
So they could predict.
Just a fixation, then after a certain point, then a face image appear, then make judgment.
Oh, so it's the state of the brain before the image appears that will determine if they can perceive it or not.
Yeah, the spontaneous activity fluctuates, sometimes up state, on down, up down. And if the picture present exactly at the up state, like by coincidence, then you can detect the image. But we have many trials, many image. Once we average them together, you see a difference for that. But the difference indicate the pre-stimulus activity.
Okay.
Yeah, thank you.
Thank you. Next question, please.
Hi, my name's Mike. I had a question for you as well. In the second part of your presentation, you discussed transitions between something. I was wondering if you could clarify.
Also attention, yeah.
Was it the trajectories of cycles that you're discussing?
The modulation is a network
transition
switch.
between trajectories.
I mean the second part, right?
Yeah.
Of my talk. It's anterior insular cortex regulate the network switch from default mode to dorsal attention.
Okay. So in the video that you showed where you could see the cycles between nodes and the connections.
Temporal circuit one?
Yeah, exactly. The
yeah.
Markov, the one that used the Markov forward assumption or something along those lines. Could you look for the transition between, could you look for transitions in something like this where in one point you mentioned that we might start to see regularities in the path that it takes, and then would a transition be like a deviation from a regularity in one of those cycles to start to seek out activity in different parts of the cortex as you pursue a different part of the task or something along?
So when you change the state of the consciousness?
Yeah, exactly.
Yeah, it's possible. You make a baseline as a model, then you see how much the trajectory deviates from your normal model when you change the state of consciousness. It's possible to visualize in that way.
So on the last part of that, would you, if you were to try to understand like a basis, not perfect basis, but sort of a collection of these paths that it could take, or the collection of regularities you could look for, do you have any experience in like collecting those things through analysis? when you're studying the different, I don't know, either the altered consciousness studies that you did, or just the basis of from the transition types of studies you did, just in terms of the collection, when you're collecting them, how to sort of parse out or detect something, or seek out a new thing to detect to look for a regularity of sorts.
The source of the regularity?
Yeah, like the different types of regularities you could see. Like with a library of words, of concepts, of whatever, like a library of paths that could be taken, or I don't know, I was wondering if you had any experience specifically with something like that.
That question is a little complicated. I don't, perhaps the regularity is in broader sense, right? But you have to, and anyway, you have to specify what kind of regularity you want to look at. So for me, it's a temporal regularity along the trajectory space, but you can also measure like entropy or complexity, all kinds of regularities.
Okay, cool. Yeah, I've been working with the time-based ones, but I think entropy studies could be a good place to look into.
Good.
All right, thank you. Thank you for your talk.
Yep.
Thank you. Next question, please.
It's a question for Dr. Huang. First of all, I just had a question about the anterior insular cortex. Have you looked at its activity during sleep, particularly during REM sleep or any type of sleep phases? Or
I haven't, because I don't have the data for now.
Okay.
So we work on participants that under general anesthesia or sedation. We haven't got the chance to study sleep state, but I would love to see the data if somebody can offer.
Okay. And just one general question. I don't know if you're familiar with the work of Celine Adesoy. I think she's done the harmonic oscillations of...
the work of what, pardon me?
Celine Adesoy, I think. She's done harmonic oscillations for different brain regions. And I guess maybe not. So, no. Yeah. Okay. Thank you.
Thank you.
Thank you. And two more questions, and then we'll have to wrap up the session.
This question is for Anirban. I'm not sure where I'm supposed to look, but he's over there.
He's everywhere.
He's
non-local.
So I'm a neurobiologist, and my research group does a lot of work in primary neuronal culture. So I was very curious about the cellular material that you're using. Can you tell us the source? Is it rodent embryonic hippocampal neurons, and how long have they been cultured?
Yeah. So can you hear me?
Yes.
Oh, okay, fine. So of course, in the papers you can find all the details, but just to tell you, we took hippocampal rat brain hippocampal neuron, which are commercially available in Lonza. We purchased it and we followed the exact prescription as they have. So we took the embryogenic cell and then we cultured. All the data are 7 days to 12 days. So we allowed 7 to 8 days to culture, to grow them. And then we started doing the measurement. But we do measurement only once, because once we take out the carbon dioxide environment, we do not put it back, as you know. And but during the measurement, we keep the whole chamber in carbon dioxide environment. Within 1.5 hours to 2 hours, we finished our measure. So most of the cells, neuron cells, were hippocampal neurons from and embryogenic cells. We start from embryogenic cells and brain hippocampus.
So have you ever considered doing experiments with mutant neurons? So neurons that either have a knockdown or are endogenously carrying a mutation, because a lot of these cytoskeletal components, their mutations are associated with developmental brain disorders in humans, and it would be, I think, of quite a bit of interest to see how those perturbations affect the measurements that your group is making.
Yeah, you are very right. That will be great. But I am a physicist, So, so what happened is in 2012, 2013, when we are publishing the single microtubule results, so everybody started criticizing us. What nonsense is this? If you measure microtubule and protein and all these kind of things, what is the guarantee that biological structures will have? So I contacted many neuroscientists to help us, but nobody was ready. Everybody was saying it is a wastage of time. Now, I would like to tell one famous dialogue I got from one of the best neuroscientists in the world. He said to me that everything inside a neuron membrane, everything inside is a fluid, is a liquid. And then at that point of time, I was in MIT, and then I heard so many comments that I recruited one postdoc, and then postdoc actually did this culture in our laboratory. So our knowledge about neurons were very limited, and then with we had the common facility where we did all this kind of thing. But my lab is very well equipped with frequencies and others. So if some neuroscientists can help us to expand our domain of this work, it will be great. So now with University at Buffalo, we are starting some experiments on organoid brain to look into, and they will be looking into the disease models, Parkinson's, Alzheimer's, and then they are working in that group. So we will be looking into the change in the frequencies, and they have built this whole infrastructure. So Professor Anirban Datta and then others are there who they have a huge team at University at Buffalo. They have got our results, so they are reproducing our results now. So they will advance it very soon. So I hope that the result that you are looking for, you get it. But if you want us very specific kind of studies, please drop an email to me, anirban.bandyopadhyay@gmail.com.
Will do. Thank you.
Thank you. That was a great question. And final question for the session.
My question is also for Anirban. So I feel like you kind of dropped the mic there at the end with some of the new findings around some of the room temperature quantum effects that you've observed within the scope of these dendritic processes. And I'm just wondering, how does that relate to some of the latest research around like biophotons and intraneuron communication in that regards? And what's your sort of hypotheses or opinion around like room temperature quantum computing? And do you foresee there being some hard evidence for the brain operating as a sort of a biological quantum computer?
So your question have several aspects of it. So let me answer one by one. The first thing that you said that the recent work on wireless communication or biophoton work. So let me remind you that as I told in my talk, there were many people who have reported on neurons and the wireless communication, electromagnetic communication since 1928. But the problem is the mainstream neuroscience never accepted. Regarding the quantum measurement, what our data is very different compared to the other data in the way is that we are exciting particular part of the biomaterial resonating at a resonance frequency, and then we are pumping the polarized light to or entangled photons we are sending at different biological structures, and then they go. What important we have got until now is that at a particular frequencies, if two proteins are excited. So these several PRL works have been published. They have talked about it, that two very distant system, they are triggered at a particular resonance frequency, and when they resonantly vibrate in a long distance, you can see the existence of quantum effect. So these similar papers are regularly published in Nature and Science. You can go, but these have not been performed on the biological systems. But if you check these papers, which are very well accepted, that if you have two large structures which are very far and you resonantly vibrate both of them at a very particular frequency, then they could be entangled even though they are not from the same source. So I think that that could be a way forward to try to understand quantum instead of electron-based communication. So that is kind of, as I told in my talk, that I'm very against of that kind of electron-based transmission, cooling it down, cryogenic temperature, and then trying to see. Instead, resonant coupling-based quantum entanglement. You just note this word, resonant tunneling-based quantum entanglement. If you search, you will find many, many papers, breakthrough works, not on the biomaterials, other sites. In that aspect of quantum mechanics and quantum computing, I think there is an opportunity for us to explore the possibilities. So right now, in my laboratory, in our laboratories, we are confirming that whatever we have got, whatever the results that we have got, we have understood. Whether it really affects in the vortex-based communication. You saw the hologram, and that is coming out of the neuron. It is a beautiful 3D, light spheres that are coming out, and then each and every component in the biomaterial have this kind of thing. And if you would know quantum mechanics, even the little basics, you know the block sphere is there. So block sphere is the kind of Hilbert space where you have all the possibilities between 0 and 1, right? So now you have many, many spheres, and that results I showed. So we have just started publishing into this coupling of Hilbert spaces, or the geometric entanglement of Hilbert spaces. So please also note this word, you can Google it, and then you'll find lots of papers in physical regulators, how Hilbert spaces could be coupled, integrated, and then geometric linking between the Hilbert spaces. So these are the two different directions, resonant coupling-based quantum information transfer and the fusion of the Hilbert spaces, the geometric coupling. These are the two directions. I think there is a possibility.
Thank you very much. I'd like to thank all of our speakers, Anirban, Zirui, Jean-Rémi in absentia. And I'd also like to thank all of you for your thoughtful questions, and how about a final round of applause? We hope to see you all tomorrow. The whole day is really going to be dedicated to altered states of consciousness, non-pharmacologic in the morning, and then pharmacologic in the afternoon, and we look forward to seeing you there. Have a wonderful evening. Take care.